Hello students, welcome back. Today I'm Anuradha from DPS Silvani Lamature going to start another topic of the same chapter, the cell unit of life, right? So in this chapter, we were discussing about the prokaryotic cell as well as the eukaryotic cell. We have done the structure of prokaryotic cells and eukaryotic cells. We were discussing about the different types of cell like plant cell and the animal cells. And if we will talk about the components which is present inside the plant cell and animal cell, we have done the outer membrane, that was the plasma membrane, and in case of plant cell we have done the cell wall. Okay, and if you will move from outside to inner side, you will find that nucleus is there. Okay, so nucleus is present and some structure which is connected to this nucleus. Nucleus is what? Nucleus is a double membrane. Okay, so here some pores are present here as we have discussed about it, and in this pore this endoplasmic reticulum is connected. Okay, we have done this thing. Endoplasmic reticulum is connected here, right? And endoplasmic reticulum was also of two types. What were they? They are the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the endoplasmic reticulum which is present towards the nucleus, it is the rough endoplasmic RER, and which is away from the nucleus, it is the smooth endoplasmic reticulum, right? So that much we have done. And if you will talk about this is a nuclear pore for the proper movement of ions. Okay, now in eukaryotic cells, they are having some more membranous structure as compared to the prokaryotic cell. So we have done that these are what? These are the endomembrane system. Along with this endomembrane system, some more membrane bound organelles are present. So what are the endomembrane system? In which there will be a coordination between some of the organelles and they are performing some particular function. Okay, so in nucleus, whatever the things which we are having like protein or the lipids, so we said that the function of RER is what? It is having ribosome. So what it will do? It will prepare or it will synthesize the protein. And if I will talk about the smooth endoplasmic reticulum, what it will do? It will form the lipids. And both of these composition are important for forming the this plasma membrane, right? So the function of RER, SER we have done. Now whatever the things we are getting here, okay, whatever the things we are synthesizing in this RER as well as SER, it will get out from the endoplasmic reticulum. So how it will move out from the endoplasmic reticulum? It will form a vesicle-like structure. What it will form? It will form the vesicle-like structure and they will get pinched off. Okay, what it will do? They will get pinched off from the endoplasmic reticulum and it will form different vesicles which contains the protein as well as lipids, right? So they are connected with what? They are connected with the endoplasmic reticulum. Now if we will move more towards the membrane, what you will see that some structures are present. Okay, some more structures are present. Okay, some more structure is what? These are the Golgi apparatus. Okay, these are what? These are the Golgi apparatus. And along with this, why we are saying Golgi apparatus? Because all these are Golgi complex, and when they are connected with this vesicles, or when we are talking about this complex as well as vesicles, it is known as Golgi apparatus, right here. So today we have to discuss about this Golgi apparatus, okay, and how it is useful for other things. It is from the endoplasmic reticulum. So all these are interconnected. That's why we are studying this Golgi apparatus under the endomembrane system. Okay. Now another point is that who observed first time? Camillo Golgi. That's why name given Golgi apparatus in 1898 for the examination point of view. Now some more important characteristics of Golgi apparatus. What you will see, these are the complex protoplasmic structure. Means that nucleoplasm and cytoplasm. So all it is combining the protoplasm. So this is a structure which is related to the protoplasm. Okay. Now comes to the another one, the many flat disc shape. So what you are watching here, that they are having some flat sac-like structure. Okay, what you are observing, flat sac-like structure in case of this Golgi apparatus, which is known as what? These are known as cisternae. So what is cisternae in Golgi apparatus? These sac-like structure which are present here. Okay, so same thing written in here, it is known as cisternae, which are stacked parallel to each other. What you can see, these are parallel with each other. Okay, now if you recall this, the structure which is present in this endoplasmic reticulum, these are tubular structure. Okay, so these are also known as cisternae. So these tubules are also known as cisternae, and these stack like structure, these are also known as cisternae. The only difference is that you can see that these are interconnected with each other. If something is coming here, it will move like this. But here, in case of Golgi apparatus, these are not that much interconnected. Okay, so this is one of the characteristic, and another one is the concentrically arranged. You can see that rings like structure. If you will see that these ring like structure is present. So what we have said, concentrically arranged, and how these are arranged? These are arranged towards the nucleus. So this is arranged where? Towards the nucleus. And if you will see more in this Golgi apparatus, you can say that it is having two faces. What it is having? It is having two faces. One is convex and another one is the concave. Okay, between this nucleus and this one, endoplasmic reticulum is also there. Okay, so I'm just explaining that this Golgi apparatus is what? These are the concentrically ring-like structure, and it is having two faces. What are the faces? Convex as well as concave. The first one is the cis phase. Okay, cis phase, it is known as the forming phase. Why forming phase? The things which is coming from this endoplasmic reticulum, it will move towards the Golgi apparatus, which is having the phase of cis form. What it is? Cis form. And if you will see the structure of cis form, it is like a convex. Okay, we know about the convex mirror and the concave mirror, like this. This is the convex mirror and this one is the convex mirror. So here if you can see the things which is present towards the endoplasmic reticulum, okay, the structure which is towards the endoplasmic reticulum, it is having what type of phase? Convex phase, and this is known as cis phase. Okay, and the structure which is away from this endoplasmic reticulum, it is known as trans phase. That's why you can see that structure is somehow this type. Okay, some cave like structure is forming here. Okay, so what is it? The phases which is away from it, it is known as trans phase, and whatever things is formed here, it get matured towards the trans phase, and it will move towards the trans phase. Same thing we have written here. Cis phase is having what type of shape? The convex shape, right? And where you can see here, towards the nucleus, okay, or towards the endoplasmic reticulum. And what it is doing? It is receiving the secretory materials. Whatever the material secreted by the endoplasmic reticulum, it gets pinched off through this vesicle and it will enter into this Golgi apparatus. So what it is doing? It is receiving the secretory material secreted by the endoplasmic reticulum with the help of this vesicle. And how these vesicles are coming here? By the pinching off. Okay. Now comes to the trans phase. Trans phase is away from it. Okay. And shape is what? Concave shape. Cave-like structure is present here. And it is present towards the membrane outer side. Okay. Then it is its role is for releasing material secreted by the cis phase. Whatever the material secreted by the cis phase, it will be accepted or it will be received by the trans phase. Okay, and here what it is doing, it is adding some carbohydrate also, because we know that along with this protein and lipids, we need carbohydrate for the proper cell. So it will come under the functions. So what we have said, it is having cis phase and the trans phase, and their functions, how it is functioning. If I will talk about the overall function of this Golgi apparatus, how can I say? It is the formation of lysosomes. Lysosome is what? It contains some of the enzymes, okay, which helpful for the lysing one. So after forming this after forming this vesicle which is coming from the endoplasmic reticulum, what it will do? It will form some lysosome like structure in which it contains the enzymes. Okay, and sometimes it also form the glycolipids and glycoprotein for the cell membrane. Okay, so we can say that the formation of lysosome takes place with the help of the Golgi bodies. Okay, now it will also act as a packaging material. Whatever the things it will be matured in this Golgi apparatus, it will get bounded by some of the membrane. That's why we are saying it will work as a packaging material also, right? Then formation of glycoprotein and glycolipids. Protein and lipids coming from the endoplasmic reticulum combinedly, and it will add carbohydrate. Okay, the complex carbohydrate has been added here, and what it will form? Glycoprotein and glycolipids, and these glycoprotein and glycolipids are useful for the formation of the plasma membrane. Because if I will recall the structure of the plasma membrane, what it is? Glycoprotein, glycolipids, okay, phospholipid. So we can say that this will helpful for the formation of cell wall also in case of plant cell, right here. So now here we will start with the another component of the endomembrane system. It is the lysosomes. Okay, as I have said, it is the nucleus. It is connected with this endoplasmic reticulum, right? And from here endoplasmic reticulum, what is there? There will be formation of this vesicle like structure, and this will send the protein as well as some of the lipid to the Golgi apparatus. Okay, it will send towards the Golgi apparatus, which is present here in this form, in the concentric form, and from here it will send, from here what it will do? It will send the structure that is lysosomes. Okay, and this lysosomes is having some fluid inside it which is acidic in nature. So now we will understand about this lipid. All these are interconnected with each other. Then in lysosome you can see that these are the membrane bound organelles, and this is the property of what? About the eukaryotic cell, that they are having the membrane bound organelles, right? So these are the membrane bound organelles which is produced by the Golgi apparatus. Same thing, the Golgi apparatus will prepare it, right? And it will remove this in form of vesicle. Okay, now these are rich in hydrolytic enzymes, or it is having the hydrolytic digestive activity, means that it is having the fluid which contains the hydrolysis enzyme, and what it will do? It will digest that cell with the help of this hydrolase enzyme. Okay, so that's why we are saying it contains the hydrolytic digestive enzyme like hydrolysis, lipase for the lipids, proteases for the protein, and carbohydrates for the carbohydrate. So with the help of these enzymes, it will break down when it will get burst out. Okay, and that's why it is also known as the suicidal bag, right? If in case of any cell, any organelle will get, we can say it will be old off. If any cell is old, what happens? This Golgi apparatus will burst itself and it will destroy the cell. In that one also it will be helpful, right? So we have said that it contains the digesting enzyme. Now this which is present inside this lysosome, it is acidic in nature. It means that the pH will be less than 7. Okay, so it is a having acidic pH, and that's why it is also known as the acid hydrolysis. Okay, now and same thing, it is capable of digesting the macromolecules. And what are these macromolecules? Like these carbohydrate, proteins, lipids. So what is the function of this hydrolysis enzyme? It converts the bigger molecule into the smaller molecule. Okay, the macromolecules get break down into the micromolecules, right? So this is the function of the hydrolysis. Now if I will talk about the function specifically, the function is digest the food content. What does it mean? If I will talk about the eukaryotic cell which is having the unicellular, means that those eukaryotes which is having the one cell, like example amoeba. Amoeba is what? It is a unicellular eukaryote. So if you will see about the amoeba, it is having what? It is having this lysosome. Okay. Or in case of any cell, if I will not take about the amoeba, if I will talk about any cell, any of the cell, what happens? That if lysosomes are present, what it will do? It will break down. What it will do? It will break down into smaller parts, and it will be easier for the cell to absorb it. Right? So same thing we can discuss about the amoeba here. In amoeba, if any of the cell, if any of the food get enter inside this amoeba, what it will do? It will start releasing some of the enzymes. This enzyme will break down this food into simpler one, and it will be helpful for the organism to metabolize it properly. Right here, so we can say that it will digest the food. And what type of digestion can be? Intracellular. It means that inside the cell only it will digest it. Right? Then it digests the old organelles also. As I have said, inside the cell, so many organelles are there which is of no use. Okay, after some times it will become the of no use. So these organelles are also get destroyed by this lysosomes, and it should be removed. Okay, unnecessary protein, unnecessary lipids, all these things should be out from the cell. So these lysosomes are important for that function. As well as if I will talk about the cell division, this one is helpful. If I will talk about the tadpole of frog, the tail which is present in the larval stage, it also get detached from the frog, or from the tadpole, not the frog. This is also due to this lysozyme, because this lysozyme is destroying that enzyme or destroying that cell which is responsible for forming the tail, or the tail cell will get destroyed by the lysosomes. This can be one of the example. Now comes to the another one, vacuoles. Okay, again vacuoles. These vacuoles mostly present in case of plant cells, right? And in animal cells also there, but mostly in case of plant cells, and they're situated at the center. It means 90% part of the cell accommodated by the vacuole. Okay, vacuole is what? Membranous sac. It means that it is having proper membrane, which is present where? In the cytoplasm itself. Cytoplasm is the matrix which is present inside the cell. Now, what is it? It stores the substance. If I will talk about this vacuoles here, in case of plant cell, if it is a plant cell and it is having what? It is having cell wall. It is having proper cell wall like this, right? So now, if you will talk about the vacuole, it is mostly in the larger part, right? So this vacuole contains what? It contains some of the fluid or store the substances. The thing which is present inside this vacuole, it can be water, it can be sap, or it can be excretory product formed by the cell. Okay, so this vacuole contains that much thing here, and the outer part is what? Outer part is cytoplasm. So the things which is present in the plant cell inside the vacuole, it will help them to provide turgidity, because it contains water. And you have seen, must have seen that example in summer. What happens? The plants get shrink. Why? Due to the deficiency of water. Okay, so what this vacuole is providing, it is providing turgidity so that it can withstand properly in the soil. Right here, so we can say that vacuole is present, and in this vacuole some structure is present here, and the outer part, the outer membrane of this vacuole, it is known as what? Tonoplast. Okay, the outer part is known as the tonoplast. As well as the function of vacuole or this structure, it will create some proper transportation system between cytoplasm as well as inside the vacuole, right? So membrane bound single membrane is present, tonoplast, in case of plant cells. Same thing written, it is covered by some membrane known as tonoplast, which is present in case of only plant cells. And what is the function of it? It will help to transport the material and ions against the concentration gradient. It means that there may be some difference for the concentration in inside the environment as well as the outside the environment. So it will maintain the balance between these two. Okay. Now comes to the another part, that this vacuole can be of two types, contractile vacuole as well as the food vacuole. What is the meaning of contractile vacuole? Same thing, if I will talk about the example of amoeba, okay, it is having some vacuole, that is the food vacuole. Okay, food vacuole is there. Food vacuole, as I have said, what is the function of this food vacuole? It will take the food. Okay, when it will take the food, what it will form? It will form some structure that is known as the food vacuole. So it is present where? Into the protozoan protist. And how it is forming? By engulfing the food particle. If it is engulfing any food particles here, so what happens here? Inside this vacuole is present, and this vacuole will, what it will do? Engulf that food and it will break down it. Okay, so it can be the food vacuole, and a digested material which is not of use, it will go outside from the cell into the cytoplasm. Okay, that much is there. And sometimes there will be contractile vacuole. If I will talk about the paramecium, okay, so they are having some structure present in their cell which helpful for the osmoregulation, because they are living in the water, so most of the water coming inside their body, so they will not tolerate that much water. So there must be a proper balance of the salt as well as water. So this vacuole, contractile vacuole, helps to maintain this osmoregulation with the outside environment and helpful in case of amoeba excretion. Okay, the undigested or unnecessary things will get out with the help of this contractile vacuole. Right here. So we have done about this endomembrane system, endoplasmic reticulum. Okay, from here, then we have started endomembrane system from where? From the nuclear membrane. Okay, from the nuclear membrane, and then endoplasmic reticulum. Then we have done the Golgi apparatus. Then we have done lysosomes, and then vacuole. Right? So we have completed here this endomembrane system. Then we will start with the another organelle. Thank you.